Lipolysis is the process by which fats are broken down in our bodies through enzymes and water, or hydrolysis. Lipolysis occurs in our adipose tissue stores, which are the fatty tissues that cushion and line our bodies and organs. Fats from muscle triglycerides and blood-free fatty acids are used for energy production. So, fat is ready and available when our glucose stores are running low between meals. And it makes sense for lipolysis to occur because it will facilitate the movement of this stored fat through our bloodstream. Fats from muscle triglycerides and blood-free fatty acids are used for energy production. There are several factors that limit fatty acid oxidation in muscle during high-intensity exercise. The reduced use of fat as an energy source during high-intensity exercise is influenced by increased levels of catecholamine hormones, which stimulate glycogenolysis in the muscles and liver, as well as increased blood glucose uptake. Increased lactate formation that occurs when glycogen breaks down and increased glycolysis flux also reduces the rate of lipolysis so that the concentration of fatty acids in the blood decreases, and the supply of fatty acids to muscle cells is also reduced. Decreased fat oxidation is related to the very minimal availability of oxygen during high-intensity exercise. Decreased fat oxidation is related to the very minimal availability of oxygen during high-intensity exercise. Elevated blood glucose and insulin levels are associated with increased concentrations of malonyl-CoA in muscle tissue, which inhibits the transport capacity of fatty acids across the inner membrane of muscle cell mitochondria. Aerobic exercise training protects against obesity by reducing body fat and chronic inflammation. Along with lipid oxidation, eating behavior, and epigenetic reprogramming, the benefits of exercise are related to increases in lipolysis and browning in white adipose tissue, and thermogenesis in brown adipose tissue by activation of the beta-adrenergic signaling pathways. Aerobic exercise can prevent various diseases if done regularly. Aerobic exercise can also control blood pressure and maintain blood sugar levels. In the long term, aerobic exercise can reduce the risk of chronic diseases, such as coronary heart disease, type 2 diabetes, obesity, osteoporosis, stroke, and cancer. Lipolysis begins when the body signals a need for energy, often during low to moderate intensity activities or when glucose levels are low. Hormones such as epinephrine and glucagon are released, signaling adipose tissue to start breaking down triglycerides. Lipase enzymes break the ester bonds holding the fatty acids to the glycerol backbone, resulting in the release of free fatty acids and glycerol. ATGL performs the first step of triacylglyceride hydrolysis, producing diacylglycerol and fatty acid. Its activity is tightly regulated by two accessory proteins, CGI5-8-NGOS2. HSL performs a second step and hydrolyzes DAG, producing monoacylglycerol and fatty acids. MGL is selective for MG and produces glycerol and a third fatty acid. Free fatty acids are transported through the bloodstream bound to albumin, a carrier protein. Fatty acids must undergo protein-mediated transport across the cell membrane into the cytosol via fatty acid-binding protein FABP. Acyl-CoA synthase then converts fatty acids into fatty acyl-CoA. Fatty acyl-CoA molecules cannot directly cross the mitochondrial membrane. The carnitine shuttle facilitates their transport. The fatty acyl-CoA must now be transported to the mitochondria through the outer mitochondrial membrane and is carried by CPTI where it becomes fatty acylcarnitine. The fatty acylcarnitine is then transported across the inner membrane to the mitochondrial matrix by CACT and converted back into fatty acyl-CoA by CPT to where it is now ready for oxidation. Within the mitochondria, beta oxidation takes place. This process involves the sequential removal of 2 carbon units acetyl-CoA from the fatty acid chain. This is the main pathway for fatty acid catabolism and occurs in the mitochondrial matrix of tissues such as liver, muscle, and adipose. Consecutive 2-carbon fragments are removed from the carboxyl terminus of fatty acyl-CoA, producing NADH, FADH, and acetyl-CoA, which are used in the TCA cycle to make ATP. NADH and FADH too, produced in the citric acid cycle, carry high-energy electrons to the electron transport chain, located on the inner mitochondrial membrane. As electrons move through the TC, they release energy, which is used to pump protons across the membrane. The flow of protons back into the mitochondrial matrix through ATP synthase leads to the synthesis of ATP in a process known as oxidative phosphorylation. Ketone levels are low during normal feeding and physiological status. They are used by the heart and skeletal muscle to preserve limited glucose for use by the brain and erythrocytes. During the fasting state, fatty acids are oxidized in the liver to acetyl-CoA, which is converted to the ketone bodies acetoacetate and beta-hydroxybutyrate. These high ketone levels also inhibit PDH activity and fatty acid oxidation to conserve glucose and allow it to enter the brain, where it can serve as an energy source. Fat oxidation during exercise. The contribution of fats to the body's energy production depends on exercise duration and intensity, training conditions, sex, body composition, and diet. At a low to moderate intensity and during prolonged exercise, most of the energy requirements for skeletal muscle can be met predominantly through fatty acid oxidation, with a small contribution from glucose oxidation. On the contrary, glucose predominates as an energy substrate during short-term intense exercise. The source of fatty acid changes during exercise. At 25% of VO2 max, the oxidized fat derives from plasma fatty acids. During exercise intensity at 65% of VO2 max, plasma fatty acids contribution decreases and the intramuscular triacylglycerol rate increases and provides about 50% of the fatty acid for total fat oxidation. When exercise intensity increases, there is a shift from fatty acid to glucose oxidation, with a reduction in the percentage of the total energy requirement derived from fat oxidation and a reciprocal increase in carbohydrate oxidation, which becomes the main energy source when exercise reaches above approximately 80% of VO2 max. Prolonged aerobic events, such as ultramarathons of 50 to 100 kilometers, is one of the examples of exercise in which aerobic lipolysis becomes the main energy system.